Okay, so this is just going to be a macromolecule review going over everything, kind of tagging on to the end of what happened yesterday in the basic macromolecule stuff. So now we're going to try to identify some macromolecules based on what they look like in their structure. So right here, number one, we have a carb, but it doesn't look like a carb. Usually carbs are found in a ring, but if we undid the chain on the ring, it would look like this. So I want to jump down here to the bottom, because number 1 and number 15 down here at the bottom are really the same, except for this has two of these hooked together in what's called a glycosidic linkage. Then we also have this one here, which is different. This would be like ribose. This is actually ribose sugar, and so it's a pentagon shape, not a hexagon shape. And it's ribose because there's an O right here. And if it was deoxyribose sugar, then this oxygen right there would actually be gone, and that's the sugar that's in DNA that connects with the phosphate. And so that kind of connects into our nucleic acids, which right here we have a phosphate, and then right here we have a nitrogenous base. So really, this phosphate would be connected up here, and then there would be bases that would come off of this, and multiple of these connecting together is what makes the DNA structure. Okay, next I want to talk about lipids, because they're different, and then proteins do a lot as well. So lipid here we can tell this is a saturated fatty acid chain because it's straight and there's no double bonds. Right here, there would be, if it was more clear, a double bond because there's no hydrogen on this side, there's no H2. And kind of what this H2 signifies is that there's the C and then a hydrogen on this side and a hydrogen on this side. And that's why it's bent here, because there's only one hydrogen, only one hydrogen. And then at the top, you have a glycerol up here, and this is actually a phospholipid, because instead of having 3 fatty acid chains coming off and forming a triglyceride, at the top we have a phosphate, and then this other thing at the top. So it's really this phosphate with all these oxygen that makes this head polar, which gives the phospholipid, this entire thing, the properties to have make the phospholipid bilayer. So next I want to talk about proteins real quick. They're really special. Let's see, this is a protein, this is a protein, and I've got some more over here. And so basically what you're going to notice on protein structure is they've got this NCC thing, which I think has a special name, but I don't know it. But this NCC with an H and OH on each end. And so what this does is that when you have, these are the monomers or an amino acid, but when they bond together, this H and OH are going to go away. And so I'm just going to fold them under so we can get the idea of it. And so the H and OH go away and they form this bond. So this bond between the C and the N would actually be called a peptide bond. And then in this reaction, water is produced because we have an H and an OH that are given off. And that would be called dehydration synthesis. Another thing about proteins real quick is that these R groups at the end. So all amino acids are the same on the top, but what differs is this part right here. As you can see, it's going to have long chains, just a bunch of different types of things that come off of it, long, short. This would be kind of hydrophobic because of all these CH bonds, so when this goes into protein structure unfolding, the stuff that's hydrophobic is going to want to move away from the water and tend to be on the inside of the protein. Or if we had a phospholipid bilayer, it would be on the inside of the phospholipid bilayer. Thanks for watching.